this here is the Flona Tapajós, so the Floresta Nacional Tapajós in Brazil, Tapajós National Forest. And this is my study site. I've been working here in various capacities since 2012. And the really large picture of the work I'm doing now, looking at bacteria that live on the surface and within leaves, what we really want to do is see if we can make a link that shows that the effects of those bacteria on leaves, or directly in terms of interactions with rainfall and the atmosphere, can affect large biogeochemical cycles. We want to know whether there's links between genes and ecosystems. And so we have this site, there's the eddy flux tower there, and we have 10 years of data of how this forest works in terms of water and gas exchange with the atmosphere. And to really address that question, the first thing we have to show is that there's significant variability in these organisms, and that variability potentially is linked to the function of leaves. So if we can scale up from the effects of bacteria on leaves and find that it has important and variable differences through time, one thing we should see in the forest is, looking at leaves, find large amounts of variability with the species that is hosting the bacteria, be it on the surface or within leaves, but also with variations of these bacterial communities through different environmental conditions. There's actually a lot of work already showing that that these bacterial and fungal communities vary with species, which is not unexpected. But to what extent they vary with abiotic conditions like light, environment, or temperature and height, those kinds of things, isn't really known. So what I did was set up a sampling campaign to simply get leaves that we knew pretty much exactly how high the leaves were in the canopy, and then also how much light they were getting. And then we process those leaves in a way where we get hyperspectral reflectance, which allows us to do estimates for basically leaf characteristics like photosynthetic capacity, leaf age, things like that. We also had subjective measures of leaf age. And so we have this big data set, and we also have preliminary results just from a culture-based study that show that at least the raw abundance of bacterial cells that you can culture on a agar plate varies significantly with both height and light. But we don't know anything really about the community diversity. And in fact, in tropical forests, there's quite a bit of work that's been done on the epiphytic portion, the stuff that lives on the surface. And there's quite a bit known about the fungal endophytic portion, the fungi that live within leaves. But there's basically nothing known about the bacteria that live within the leaves, in tropical leaves, or the relationship of the total microbial community, both fungal and bacteria, between the inside and the outside of the leaf. So we sampled in a way where every leaf sample we have collected both the surface bacteria and fungi and the internal bacteria and fungi. But to look at diversity, we're not doing culture-based, we're doing non-culture-based, and that's where it's been a kind of a slow process to get this number of samples, do the DNA extractions for this number of samples. So that's my main purpose that's being funded for this trip, is to be working in a lab, not in this beautiful forest, which I did earlier, climbed up these trees to get the samples, but working in a lab to extract close to 800 DNA samples from either leaf material itself or the kind of leaf funk that came off as a wash. And to understand a little bit about what we're talking about here, here's a real leaf, and you can see here's some younger leaves that are quite clean, or relatively clean. And this in the tropics is pretty common for an old leaf to be covered in all kinds of weird epiphytic stuff. And you can also see colonies that are probably somewhat internal to the leaf as well. And on the underside of the leaf, it's not quite as notable, but you can see weird brownings, etc. So leaf age is also going to be an important component that we have ways to try to incorporate in our measures. So at the very least, we're hoping to, or as a start, we are hoping to find what this variation is of bacterial communities along height and light gradients in just 4 big emergent species. One mid-canopy species that's the dominant mid-canopy species, and one shrub in the understory, which is the dominant understory shrub in this ecosystem. And so that's the main goal. At the same time, I've been working a lot with my partners here. So this whole site is part of what's called LBA, which is the Large-Scale Biosphere Atmosphere Interaction Project that started with NASA and the Brazilian government, is now mainly run by the Brazilian government and foreign research partners like my lab group. And then, of course, the university in Santarém, where I'm working with a lab group that they're not so much interested in ecology of these leaves, they're interested in isolating actinobacteria from these leaves, because actinobacteria often have very important and useful, either industrially or medically, secondary metabolites. So that's where we paired up, is I wanted a rough count of the raw abundance of culturable bacteria, and they wanted just to isolate new actinobacteria. And then at the same time, we're making a big exchange, more or less in skills. So I've been working with their lab group to teach various members how to do DNA extractions and start them up on more or less doing 16S sequencing of a bunch of previously isolated bacterial strains they have. While at the same time, we got about 9 new actinobacteria as endophytic bacteria and 9 epiphytic bacteria that a number of undergrad students, one undergrad student who's now going to be starting the master's program at UFOPA, have been working with as part of their main research. In terms of potential policy implications of this sort of research, one is just understanding whether these organisms play important roles in forest function. But then also, of course, whether, if they do, how that may be affected by climate change or affect climate change in the future. Expert tree climber, beware, beware.